So in Lab 6, we are going to be looking at transport, specifically diffusion and osmosis, as we are looking at how things can move through a plasma membrane of a cell. So we are going to be looking at much of this from the terms of a cell, since we are talking about living things in this class, but we are going to have to mimic that a little bit because we can't always use living cells. So we are going to use what's called a dialysis tubing that is going to act as a plasma membrane. So as usual, you will see in the first little part of your lab book your objectives and keywords that you should be paying attention to, learning, understanding. Basically, you should be able to identify and answer all of these objectives at the end of this lab so that you are prepared for your assignments and quizzes and of course that lab exam. So pay close attention to these. We're going to be walking through this right now as well. So let's start by reviewing our plasma membrane. So as you were talking about cells in your lecture component of this class, you learned that all cells have a plasma membrane, right? Literally every cell, whether you are a prokaryote or a eukaryote, you have a plasma membrane. So that plasma membrane has a few components. One of the most prevalent in our plasma membrane are called the phospholipids. So phospholipids are lipids that are both hydrophilic and hydrophobic. So you can see in this picture here, the red little dots up here are the hydrophilic heads, and then the little yellow strings off of them are the hydrophobic tails. So because of the way phospholipids interact with the watery environment in living things, the hydrophilic heads will be on the outside of the membrane as well as the inside of the membrane, so like on either side of the membrane. And then you will have the hydrophilic components kind of facing towards each other. And this gives us a boundary, a very good living boundary. Now you'll also see in here in this picture and what you saw in lecture, there are proteins, which in this picture are the larger blue proteins that are in here. And there's little cholesterols, which you can see the little yellow components here, which are fatty based. So they'll be in the center-ish of our plasma membrane. And then you should see some glycocalyx, which are the glycoproteins and lipids that are going to be attached to the outside of the cell, at least in this picture. They're on the plasma membrane, which are going to help with things like identifications. So the plasma membrane, one of the really important things you should realize about the plasma membrane is that it is always changing. Since it is always able to change, it can always adapt to the needs of the cell. That's why we call it a fluid mosaic. This is the fluid mosaic model or fluid mosaic theory, that we can remove proteins if we need to. For example, we can put in more cholesterol if we need to. So the cells can adapt to what's going on around them. This also means that they are what we call selectively permeable. Selectively permeable means exactly what it sounds like. Some things can move through it, some things cannot. So because of the makeup of our plasma membrane, small things can move through the membrane very easily, especially those that are not charged. So for example, gases can move through the membrane. Oxygen and carbon dioxide pass very easily through the plasma membrane. And also hydrophobic things, because again, there's no partial charges on those molecules. So things like steroids can move through the membrane very easily, as where the bigger molecules and or the charged molecules cannot pass through this membrane easily. So things like water, small but pass through a little bit, but actually need a protein to help them do that. Things like ions, right? Sodium ions cannot pass through this membrane easily by themselves. So yes, these things can still move through. So the hydrophilic molecules and larger molecules can still pass through the membrane. They just need some help to do that. So you will see in this picture as well, there's a protein channel. So this channel may allow for ions to pass, for example, may allow water to pass. So each protein channel will have its own function. It will have its own job to pass things in or out of the cell. So it's not that nothing can pass through this membrane. It's just that the cell really controls what can move in and out of this membrane to keep it consistent. So in our lab, like I said, we're going to be using what's called dialysis tubing. Now, dialysis tubing obviously is not alive. It is plastic, but it has little holes in it. And those holes are going to mimic what we see with plasma membranes. But basically, the dialysis tubing cannot differentiate based upon charge, but it can differentiate based on size. So the smaller items can move through this dialysis tubing, the larger things cannot. So that's kind of what we're going to be studying and looking at in this lab. So let's talk a little bit about movements and solutions. So solutions are going to be mixtures of different things, right? Different materials, molecules that are going to sometimes be homogeneous, sometimes not. So a homogeneous solution means that it is the same throughout the entire mixture. The dissolved amount of solute and solvent is the same throughout the whole thing. So when we think one of the examples I always hear is like Kool-Aid. When you make Kool-Aid, you mix in the powder and the water and you have a homogeneous solution. In living things, we also, like our blood in humans is pretty homogeneous, right? You can sample it from just about anywhere and it'll be about the same. So all solutions are going to be made up of solutes dissolved into solvents, right? So in that Kool-Aid example, the solvent is the water, the Kool-Aid powder is the solute. The solute will be dissolved in the solvent. If you can do that so that it mixes all the way through, it becomes a homogeneous solution. So when we look at solutions in living things, a lot of times we talk about this in relationship to water, right? That the solvent is the water. That is true of hydrophilic things, right? Those that love water, but it is not true of hydrophobic things. Hydrophobic materials cannot dissolve well in water, but they can dissolve into other hydrophobic things. So water is cool. It's great, especially with living things. It is a great solvent, but it's not the only one. It doesn't dissolve everything. So in this lab, you are going to be looking at passive transport, which means there is no energy that is actively needed right now to move molecules around. So diffusion and osmosis are examples of passive transport. So again, no energy is needed right now in order for those molecules to move, right? They have kind of stored up energy in order to move. Active transport, on the other hand, needs active energy right now. It is actively burning energy. One of the classical examples of this is how we use ATP, which is adenosine triphosphate, our energy molecule, to move ions across the plasma membrane when we talk about muscle contractions and how our brain sends messages when you get into your anatomy classes. This is called the sodium-potassium pump. So the sodium-potassium pump is going to use energy to push things against their concentration gradients. So passive transport will allow molecules to kind of move by themselves following their concentration gradient. Active transport is going to be forcing things against their concentration gradient. So in this picture, we're going to take sodium, where there's only, so sodium's these little orange ones, 1, 2, 3 of them down here, and there's like 12 of them up here. I didn't count them. And we're going to use this pump to push them up so that there's more outside here than there is inside. This is setting up a better concentration gradient. So passive transport, no energy is actively needed, and we go with the concentration gradient. Active transport, energy is needed, and we have to go against the concentration gradient. So what is the concentration gradient? So the concentration gradient is going to be the difference between concentrations, how high one side of the membrane is and what we're talking about, and how low it is on the other side. The higher the difference, the bigger the concentration gradient, the faster we're actually going to see molecules move. So one type of passive transport is going to be diffusion. So diffusion is going to be the movement of a molecule of some kind down its concentration gradient until we reach an equal distribution. So we are going to move from a high concentration of that molecule to a lower concentration of that molecule. So in this picture, we have a drop of dye that was dropped into water. Now the dye is able to dissolve into water because it is hydrophilic. So as it passes through the water, you can actually watch it move from a very high concentration in the center where the drop started to lower concentrations all the way throughout. And this would continue until this entire glass of water was red. That is diffusion, the movement of the red dye from a high concentration in the middle of the glass to a lower concentration elsewhere in the glass until the whole glass is red, until we reach what is considered an equal concentration.